Welcome back, everybody. We're going to finish up chapter 1 strong, and we're going to start out talking about the approach to studying microorganisms. So we use a systematic approach, and the first thing we're going to talk about is taxonomy. So taxonomy is classification, description, and identification of organisms. It's a mechanism so that we can name organisms, group them, and name them. So this first, the taxonomic system was developed by Carlos Linnaeus, and this was in the 1700s. So the original groups were plant and animal. Those were the only two groups that individuals knew of. If you were movable, then you were a plant, or if you were movable, you were a plant. If you were movable, you were an animal. If you were stayed still and you photosynthesized, you were a plant. He used binomial nomenclature to name organisms, so to give them a specific name. Binomial nomenclature uses the genus and the specific epithet of an organism to come up with their species name, and so that's Homo sapiens for humans. A phylogenetic tree is a way of showing how organisms are related to one another through evolution, and so this is a beautiful phylogenetic tree that's probably not very accurate today, but at this time it was relatively accurate. I'll just let you know that phylogeny is constantly changing. So taxonomy is always getting changed and moving around. Different organisms are put in different groups all the time. So this is Ernest Haeckel's rendering of the Tree of Life. And this was from the book, The General Morphology of Organisms. And he had 3 different kingdoms. So instead of just having plant and animal, he added protists. And later he added another kingdom called Monera. So these were for unicellular organisms that didn't have a nucleus. So basically your prokaryotic organisms. So we have Linnaeus with the 2 kingdom system, and Ernest Haeckel added Protista and Monera. And look at the dates that he was alive. So this is relatively new. Then Robert Whittaker, in between 1920 and 1980, that was when he was alive, he added an empire. He called it the Prokaryote Empire. And that empire only housed Monera. The empire Eukaryote, sorry, dogs, I have dogs. So if you didn't know, I have dogs and they're evil. My dogs are wonderful, but they do bark when people are anywhere. So sorry about the dogs. I do try to be or make these videos very effective, and I guess sometimes having a dog bark will wake you up if you're getting bored with it. So on that side, you know, yay, congrats for waking back up when my dog barked. Anyways, let's go back to what I was talking about. We have Carlos Linnaeus, who in the 1700s had made a 2 kingdom system, plant and animal. Then Ernest Haeckel added Protista and Monera. Protista, unicellular organisms with a nucleus. Monera, unicellular organisms without a nucleus. Robert Whittaker then added 2 empires, Prokaryote Empire, which only contained Monera, no nucleus, and then the Eukaryote Empire, which contained Protista, plant and animal, and fungi. So now we're adding fungi into the mix. All of these organisms have a nucleus, whereas monerans have no nucleus. So the term karyote is nucleus. U is true. So basically he said empire true nucleus, these guys, and empire pre-nucleus, these guys. Okay. So at this point, up until this point, we were basing everything on observable characteristics. So what can we see? What do we notice about these organisms? Are they using carbohydrates? Are they photosynthesizing? Are they walking around, or do they squirm around? Do they stay still? Those were characteristics that we could observe. Then Carl Woese and George Fox, they took what we were doing up a notch and started genetically basing phylogeny. So they started looking at what organisms' genes look like and putting them into different groups. Okay. And so their phylogenetic tree contains 3 different domains. So prior to this, we had the 2 large categories, eukaryote and prokaryote, and they came up with a 3-domain system, which included your eukaryotes all under the header of eukarya domain. And then the prokaryote groups were split into 2 different subgroups or domains, bacteria and archaea. And they looked genetically at all of these organisms, and so you can see where we are, we're animals, and they put them in a phylogenetic tree, and they noted that bacteria have less in common to eukaryotic organisms than archaea. Archaea are more closely related to bacteria if you look at where they are together, but they're definitely also related to eukaryotes. Whereas eukaryotes and bacteria are very far apart on that phylogenetic tree. So here is a systematic approach of identifying humans. So we all fit under the domain Eukarya. We are all in kingdom Animalia. We are animals. Phylum Chordata, we have a backbone or a spine. Class Mammalia. So we are mammals. We have hair on our bodies, and we breastfeed our young. Order Primata, which means we are primates, so we can walk on 2 feet. Family Hominidae, and then genus Homo, and species Homo sapien. Okay. But in this class, we're going to be focusing more on bacteria and microscopic organisms, not on humans. If you want to learn more about humans, you can take anatomy and physiology. I teach it every fall. I teach AMP1, and every spring I teach AMP2. So you can always join my classes. So in microbiology, though, we have a very important book that people always use when they are trying to identify microbes. So this is known as Bergey's Manual of Determinative and Systematic Bacteriology. This is a standard reference book for identifying and classifying bacteria. Bacteria can be identified through biochemical testing, through genetics, and through serological testing, so antibody testing. So they have a lot of mechanisms for testing bacteria, and so we can put bacteria into different groups themselves, actually. There we go. When we look at microorganisms, microbes are super small. So when we say microorganism, we are dealing with living organisms. But when we use the word microbe, then we can also include non-living infectious agents. And so I typically say the word microbe when I'm talking about microorganisms, just because that includes both bacteria, viruses, prions, the whole scheme of everything, okay? But these organisms are too small, or these materials are too small to be seen without a microscope. Some fungi and some parasites can be seen without a microscope, but a lot of times the initial structures are not. So like the the parasitic eggs are not able to be seen without the aid of a microscope. And fungi, a lot of their toxins can't be seen. Their spores that they produce can't be seen. And so we study them in this class, specifically associated with what type of diseases they might cause and stuff like that. So when you look here you can see things that are very large. A frog egg is super large. A lot of times you can see frog eggs. They're large enough to be seen by the unaided eye. Not always. So once we get down here though, a human egg or pollen, these are super small and most people cannot see them with an unaided eye. Once we get down to the cell level, you can't see these with an unaided eye. I'm sorry. So you need a microscope. And even with the frog egg, you can see them possibly, but a microscope will allow you to see them more effectively. Blood cells, they are large, but they're still too small to be seen by the unaided eye. Microbes like bacteria are going to be only seen using a microscope. Organelles, and then we look at viruses also using a microscope. Some viruses are so small that a typical light microscope, which is what we have in our lab, doesn't work effectively enough, and so you can't see them unless you have an electron microscope. Prokaryotes, then, including bacteria and archaea, are organisms that lack a membrane-bound nucleus. So these organisms are prokaryotic pre-nucleus. Archaea are prokaryotes that are unicellular, and they can be found in almost every single environment. If you look at bacteria, bacteria are prokaryotes that are unicellular and also inhabit nearly every single environment. The difference between the two is in the cell wall surface. Bacteria have a substance known as peptidoglycan, which I will write down or you'll see in a future slide. But peptidoglycan is a substance only found in bacterial cells, whereas archaea have a different substance known as pseudopeptidoglycan or pseudomurine. That's how we separate the two groups. Archaea are also non-pathogenic. We have not found any pathogens in the domain archaea, whereas bacteria have both beneficial and some pathogenic strains of bacteria. Under the eukaryotic group, we have algae. Algae are plant-like organisms that can be unicellular or multicellular, and they derive energy via photosynthesis. We have protozoans. Protozoans are always unicellular. They do not have a cell wall, whereas algae have a cell wall. And protozoans typically are motile, but not always. Protozoans are probably our craziest group of organisms, and they can get really confusing when you're trying to put them on a phylogenetic tree. Fungi include both molds and yeasts. Molds are multicellular, yeasts are unicellular, and they can be very beneficial. We use molds a lot of times when we eat foods. Yeasts help us to ferment certain types of beverages like alcohols, and also help us to make breads and other substances. And then we study helminths. Helminths are multicellular organisms, and they can sometimes be very large. They definitely, a lot of times, can be seen by the unaided eye, but their eggs and larvae are often microscopic, and because of that, we study them here in the microbiology field, as well as in the field known as parasitology. So this is a picture of your protozoans. These are different protozoans. They're called diatoms. Diatoms are very beautiful and typically don't cause any problems unless they start secreting or unless they start growing out of control and secreting certain toxins. Here we have a protozoan. This is Giardia lamblia. Giardia lamblia is a protozoan that infects humans when we drink infected water. So these guys cause severe diarrhea. This is Candida albicans. So this is a fungus. Candida albicans is actually a very common agent in the vaginal region of females. Well, okay, there's no vagina in a male, so that makes sense. And the vagina, but if it grows out of control, like if you are taking an antibiotic that kills off the lactobacilli, then a lot of times that will cause the candida to be able to grow out of control and cause a yeast infection. It can also cause oral thrush. That is typically seen in infants. So this is just fungus growing on fruit. Fungus can be very beneficial, but it also can cause a lot of food spoilage. And so that's what this picture is depicting, is food spoilage. And then here we have our multicellular helminths. So this is a tapeworm. This is a special worm known as the New Guinea worm, or Dracunculus medinensis. So this beef tapeworm, known as Taenia saginata, can infect cattle and humans. The eggs are microscopic, but if a human gets an egg in their system, that egg can hatch, and then the worm, because it is it has both male and female sex structures, it is able to reproduce with itself, and so it can basically fertilize its own eggs and grow out of control. This worm here, Dracunculus, can only get into your system if you drink water with the worm in the water. So the worm is typically taken in by a crustacean, and then you drink the water, and the crustacean, which is a microscopic structure, gets into your system. The worm will then move down your body to a low region like your foot, which is what we're seeing here, and will so it'll grow, it'll move down your body, and then just the butt end of the worm will come out, and it will lay eggs when you walk into water. The only way to remove this worm is by rolling it around some structure. Every day you have to pull it out little by little. You don't want to break it, because if you do, the toxins that are going to be released in the body would kill the person. And that's why you also can't use antihelminthics against this worm. We are done with this chapter. So I will post this video, and we'll get the next videos for chapter 2 running, okay? Bye.